Hello and welcome to IB Bio 1.2 Notes. So we'll be starting off with talking about prokaryotes. So prokaryotes are the earliest type of cells, and they include two types. You have bacteria and archaea, and they're simple unicellular organism, meaning they're only composed of one cell, and they have no compartmentalization, meaning none of their organelles are bound by membranes. So all the metabolic processes happen inside the cytoplasm. So the organelle structure and function. First, we have the cell wall. We can see that right here. And the cell wall encloses and protects the cell. It maintains its shape and prevents it from bursting in a hypotonic situation. And then we have plasma membrane, which is right here. It surrounds the cell and it controls the movement of substances from the outside into the cytoplasm on the inside. And so the cytoplasm is a medium that fills the cell, and it's where all the metabolic reactions occur in prokaryotes. All right, next we have pili. So these little structures outside, they're protein filaments on the cell wall, and they help in cell adhesion, which is their interaction and the transferring of the DNA between two different prokaryotic cells. After that, we have the flagella, which is this long whip-like structure, and it just basically helps the prokaryote propel or move forward. So, and then we have the 70s ribosomes, which are the sites of protein synthesis. 70S, right? And then we have the nucleoid region, which we could also call naked DNA or nucleate DNA, and it just controls the activities of the cell and the reproduction of the organism. It's where all the genetic information in a prokaryote is stored. And then we have plasmids, which is this small circular structure, and they carry a few genes which often give the cell antibiotic resistance. So you're going to need to write all of these organelles down, and you're also going to have to know how to draw and label a prokaryotic cell, as well as identify the organelle structure. So this is a drawing of a prokaryotic cell, and you could just follow this through to remember how to draw it. And then, so 70S and 80S ribosomes are different. 70S is in prokaryotes, and they're smaller than the 80S ribosomes that are on eukaryotes. And so we know that ribosomes are what produce protein. And then we have prokaryotes reproduce through binary fission, which is basically they split into two daughter cells asexually. And the process first starts when chromosomes are replicated semi-conservatively in the beginning, at the point of origin. So you're going to have to use these words when you describe it on the test, semi-conservatively. And the second part is that the two copies of the DNA move to both ends of the cell and attach to the membrane. The cell then elongates, and finally cytokinesis occurs, in which the plasma membrane inches inward and two daughter cells are formed. Alright, now on to eukaryotes. So eukaryotes, unlike prokaryotes, are organisms with complex cells in which the genetic material is located in the nucleus and is isolated from the cytoplasm by the membrane. So eukaryotes evolved through endosymbiosis, which is basically a process in which prokaryotic cells engulfed each other. And that's not important. They all have compartmentalization, which is the formation of compartments within the cell by membrane-bound organelles. So you're going to have to know the definition of compartmentalization as well as its advantages. So the first advantage is that metabolism has a greater efficiency, since enzymes and substrates are more concentrated since they are in an enclosed area. The second advantage is that all the internal conditions, such as temperature and pH, can be different for each specific organelle. The third is that it isolates toxic or harmful substances away from the cytoplasm and into specific areas. And then the fourth is that the flexibility of changing the numbers and the position of organelles within the cell, which cannot happen in prokaryotes. Alright, so in eukaryotes, these are the organelles. So we have the plasma membrane. So first of all, there's two types of eukaryotes. We have plant cells, right here at the bottom, and an animal cell at the top. So they're mainly the same except for a few differences, but they both have a plasma membrane which just controls the movement of substances in and out of cells, so the same as prokaryotes. Then we have the cytoplasm, which is also the same as prokaryotes. It fills the cell and holds all organelles, and it also contains enzymes that catalyze reactions. Then we have the mitochondria, which is not in prokaryotes, and it's the site of cellular respiration, where ATP is produced. After that, we have ADS ribosomes, which are the sites of protein synthesis, so these small red dots right here. And the free ribosomes produce proteins that are used inside the cell itself. We then have the nucleus, which are not in prokaryotes, right here, this big circle, and it just controls all activities of the cell, as well as reproduction of unicellular organisms. We have inside of that the nucleolus, which is the part of the nucleus that produces these ribosomes that each go on to produce protein. Then we have chromatin, which is basically the part that contains all the genetic material of the organism, which is inside the nucleus. It'll be around here on the inside. And then we have the smooth endoplasmic reticulum. So we could see that right here, this green thing does not have ribosomes on it, and its function is to make and store lipids, such as steroids. Then we have the rough ER, right next to the smooth ER, and this one has a lot of ribosomes on it because it transports the protein made by those ribosomes to the Golgi apparatus right here, which is what later ships them to be used outside the cell. So these are the drawings of the animal cell at the top and the plant cell at the top, so you're going to have to know how to draw them and label them, and also memorize the structure and function of the organelle. So the Golgi apparatus, what we said before, is its function is to process and package proteins to be released into vesicle, which then takes these proteins outside of the cell. And the vesicle is a small sac that transports substances outside of cell, and it does so by fusing with the membrane right here. And lysosomes, so these are only in animal cells. These are actually not in this picture, but lysosomes they contain enzymes and play an important role in the destruction of waste and old organelles. Then we have centrioles, which are also not here. So centrioles play an important role in the process of nuclear division, so the splitting of cells, by helping to establish microtubules, which we'll discuss in another section. Then we have the vacuole, so this is actually found in both plant and animal cells. So plant cells have a larger vacuole, and it helps with the osmotic balance of the cell, so the water balance, and in storage of certain substances. Then we have the cell wall, which we can see right here. It encloses and protects the cell and helps maintain its shape, and also prevents it from bursting in a hypotonic situation. Finally, we have the chloroplast, which is also only in plant cells, just like the cell wall, and it is a double membrane bound organelle that contains chlorophyll and is mainly responsible for photosynthesis. Okay, the next section we have is electron microscopy. So microscopic resolution is the shortest distance between two separate points in a microscope's field of view that could be seen as distinct objects. So you're going to also have to memorize this definition. So electron microscopes have a much higher resolution than light microscopes, which is why we could actually label the organelles and structures within the cell. So electron microscopes have a resolution of 0.1 nanometers, while light microscopes have a resolution of 200 nanometers. So that's just the difference in resolution. Electron microscopes have no light, while light microscopes you can see the light of what's under there. And so you have to know also how to analyze electron micrographs and identify organelles, as well as deduce the specific function of specialized cells. So for example, this would be the nucleus. Right here, this is the nucleolus, and so on. Yeah, that's it. Thank you.